So we saw an increase in these whole body stereotypies in a number of different dyadic comparisons. We saw them, first of all, doing more of these stereotypies while they were just hanging out solo in their cages. We also saw them increased in familiar pairings, that is, when a monkey had a cage mate that it knew quite well, and also in the unfamiliar pairings. And just to show here that the control animals, the untreated controls, and the controls that had the antibodies from mothers of typically developing children did not show these whole body stereotypies. So conclusions from this pilot study are that although we didn't see any pronounced differences in social and or communication behaviors, we did at least see one sign of a core behavior in autism in these monkeys, and that was motor stereotypies and hyperactivity. So as I've been saying, this has been a pilot study. There were only 4 monkeys in each group in this study. Again, monkey studies are very expensive to conduct. And currently in the lab, we're trying to replicate this finding, and we have a new and improved maternal antibody model. This time we have 32 subjects, which actually is quite a lot of subjects for a non-human primate study. And they're divided into 4 experimental groups. We have the replication group, where they're receiving injections of the purified antibodies at the same time points. We also have a targeted, so in the previous study, they actually had antibodies purified and blended from all the mothers who had mothers of children with autism. In this study, we're actually isolating that 3773 band and looking at them as their own group. We have, again, the typical IgG control group and then another group of untreated controls. Here's the timeline of exposure to the IgG antibodies during pregnancy. Again, the rhesus monkey gestation is 165 days, roughly divided into 3 trimesters. In the pilot study, they received 3 injections at 27, 41, and 55 days. And now in the current study, they're receiving 6 injections total, spanning from late first trimester into early second trimester. So as we watch these monkeys grow up, there are a number of behavioral assessments that are going on. In the first 6 months of life, which is roughly equivalent to 0 to 2 in a human, there are twice daily home cage observations and social group observations as well. Here's a timeline of their first 6 months and the different specific tasks that they're introduced to. We are testing imitation at birth. This is really fun, to stick your tongue out at a monkey and see what it does to you without cracking up. The human intruder task, where a human walks into the room and the monkey is evaluated to see how they respond. Typically, they don't really like it. And other tasks as well going on. What I think is really interesting is we've now added an MRI component to this study, where we're imaging these monkeys at multiple time points: birth, 1 month, 2 months, and 3 months. And then on between 6 and 12 months, there's 2 additional time points for MRI. Here's just showing the behavioral assessments that are being conducted right now in the 6 to 12 month period, and then the MRIs are being carried out at 6 months and 12 months. So that is actually about all I have to say about the monkey model right now. As I said, again, monkey experiments take a long time. We're waiting for the monkeys to grow up. Rhesus macaques are seasonal breeders, so we had to wait to get the whole colony pregnant. And the subjects are now just turning 1 year old. So hopefully next time, David can come back and give a presentation on the results of that study. The implications for the study, however, I think are very, very interesting. This maternal antibody model provides the possibility that there might be a diagnostic marker for a risk factor for autism. In 12% of these mothers who had multiple children with autism, they had these antibodies. What's even more exciting is that there's a potential for intervention here as well. Mothers could have plasmapheresis to actually remove the bad antibodies and perhaps prevent the autism from happening. This is all, of course, just a dream right now. So now going on to the second approach that our lab is taking. And this is with the Autism Phenome Project. It's really a Mind Institute-wide study going on. It's a large-scale study with a target enrollment of 400 children. The age of enrollment is 2 to 3.5 years of age, and these kids come in for 5 different visits. They get a whole battery of testing done to them. So it's really kudos to the families and the children for participating in this study. But actually, we have families contacting us from around the U.S. wanting to participate. These kids have an extensive behavioral battery that's led by Sally Rogers. We take extensive medical histories. We're taking blood draws from both the parents and the children, as well as affected siblings, to look at immune factors and genetic factors involved. And then where I come in is we're looking at brain structure, and then we also have an ERP team that's looking at brain function. The whole goal of this study is to try to identify different subtypes of autism. And what I'm going to show you here is just a couple of the preliminary findings that we've seen between brain structure and the immune system. But first, because this is what I do, I wanted to show you how we acquire the MRI scans in these children. If any of you have ever had an MRI scan, you might wonder how we get a 2-year-old with autism or even a 2-year-old with typical development to undergo an MRI scan. And this, in fact, is what David had asked me to do when I first started in the lab, and I thought that my postdoc would be very short because I didn't think that I was going to be able to do this. But what we did was create a child-friendly scanning environment. Here's a picture of our scanner. Oh, sorry. As it is. And we worked really hard and collaborated with Dr. Rogers to create a pediatric-friendly environment. So what we've done here is we've disguised all of the sort of medical scary things in the room. We put 2 gurneys on either side of the MRI bed so that there's a nice big family bed where parents can get up there and lay down with a child. We do all of our scanning while the child is sleeping. And so getting the child to fall asleep is the biggest trick. We have a weighted blanket that we use to cover the child once they're asleep, and that helps them keep snug and on their back. We do things like block off the computers in the control rooms so that the children don't get excited by them and not want to fall asleep. And then during the scans, we have research assistants that stay with the child for the entire duration of the scan. We invite the parent to stay in the room with the child as well in case the child wakes up during the scan. And we've had incredible success with this approach. Our MRI protocol is actually quite comprehensive, but we order it in a sequence of priority where our T1 weighted structural scan is the most important, and it's our first sequence. We're also doing diffusion tensor imaging in these kids, and about a year ago we added a bold sequence so that we could look at resting state functional connectivity. Of course, these kids, when they're sleeping, can't be doing any task-based fMRI, but functional connectivity is actually a very interesting and exciting area of research in autism, I think. We also collect T2 weighted images for clinical reasons, and the other things are more for quality control. So our success rate thus far has been about 90%. When we started this study, we were told by other colleagues who do sleep imaging in young kids to expect about a 50 to 60% success rate, and we've actually surpassed that quite well. We've scanned over 200 children using this method, and moreover, they've returned for longitudinal imaging at 2 different time points with 1-year intervals as well. So I think we're on about 120 kids who've come back for their second time point. And the third time point is getting a little bit harder because they're getting a little bit too old to sleep through the MRI scans, but we're still acquiring quite a bit of that data. So here's the demographics for the subjects for the time 1 scans. We actually have more because I'm sure they collected a scan last night, but we scan every night of the week. And what I want to show you now is some of the preliminary findings that we've been trying to work out with the APP, in conjunction with the APP immune team. So from the blood, they're getting cytokine levels and a whole host of immune profiles from these children. And this is, again, with Dr. Vandewater and Paul Ashwood. So one of the findings that they were interested in looking at was looking at adhesion molecules. And these adhesion molecules play an important role in inflammatory processes. Specifically, we looked at a number of them, but there was one that we were interested in called PCAM1. because there was a previous study showing that PCAM1 levels are decreased in individuals with autism. And moreover, that study found that PCAM1 levels were negatively correlated with head circumference at birth. Why this is, we don't know, and we still don't know. But just to show you what we found in our sample, we replicated the finding that PCAM1 is reduced in the children with autism. And when we looked at total cerebral volume, we found a strong correlation with total cerebral volume and PCAM1 levels in the typically developing children, but we didn't see any such correlation in the children with autism. So the kids with autism are the red dots, and the blue circles are the typically developing. So this is MRI at age 2 to 3.5, and this also correlated with birth head circumference, or the same relationship held true where the typically developing children at birth, their heads were already correlated with their PCAM1 levels and not in the children with autism. This is the sort of finding that we're excited about that's coming from the APP, because who else would think to look at the relationship between PCAM1 and brain volume? Where we can do it, and then the next step would be to explore the mechanism underlying why this is. Another sort of brain immune finding that we've been looking at is looking at frontal lobe subgroups. So many of you know, and Dr. Minshew just mentioned, that there's this total brain enlargement in young children with autism. Well, it turns out that the frontal lobe is driving much of this brain enlargement. So here's just plotted age and frontal lobe volume. And what I want to draw your attention to is that here is just a confidence interval for the typically developing children. And then there are these kids with autism who have either extremely large frontal lobes in the green and a smaller subset in the purple that have sort of smaller frontal lobes. And the whole point of the APP, again, is to subgroup children. So what we're interested in looking at is if there's anything different between this big frontal lobe group and small frontal lobe group. And another thing that we found in collaboration with Judy was a relationship between IP10, which is a pro-inflammatory cytokine. Interestingly, though, we saw that the children in the small brain subgroup tended to have these higher levels of IP10. And again, this is a finding that we're going to be exploring and looking into further, perhaps looking at their genetic influences as well. So I've shown you some preliminary evidence suggesting that there are specific brain-immune relationships in children with autism. We're going to continue searching for immune-related subgroups. One thing that I'm interested in looking at is, now that we have over 200 moms in the APP sample, we're actually replicating the finding that about 12% of them show this abnormal maternal antibody that I talked about in the non-human primate model. Well, I suggested to David, instead of spending all this money and looking at a non-human primate, why don't we look in the kids in the APP sample and see what their brains look like and if they have a different developmental trajectory. We're also going to continue cross-disciplinary analyses to explore these specific immune-related subgroups. So that's it. I just want to acknowledge everybody involved. The maternal antibody model is really led by Melissa Bauman, and the only reason I could talk about that is because we've been best friends since grad school, so I felt comfortable presenting her research. And then the Autism Phenome Project, a whole bunch of people at the M.I.N.D Institute are involved in that as well. Thanks.